For the next oral presentation is a directed evolution-derived chimeric receptor displaying a natural killer receptor, Repertoire, exhibit pan-cancer cytotoxicity. Presented by Xudong Tang from Naval Medical University, which will be done online.
So can everybody hear me and see me? Okay. And now let me start my presentation. Okay, dear professors, good afternoon, good noon. It's my great honor to talk about my project, a direct evolution-derived chemical receptor displaying a natural killer receptor repertoire exhibits pan-cancer cytotoxicity. And first, background. And CAR-NK embodies a cutting-edge approach for cancer treatment and mainly pathological malignancy at present. However, tumor microenvironment is characterized by suppressive cells, physical barriers, and overexpressed checkpoint. And apart from these, cytokine deficiency and tumor heterogeneity occupy a larger proportion. So, addressing cytokine deficiency. In tumor microenvironment, inhibitory cytokines like TGF-beta are abundantly secreted by suppressive cells, while essential activating cytokines like interleukin-2 and 15 are scarce. And altogether, the condition hinders the activation and proliferation of NK cells. And next, heterogeneity. So when single-targeted CAR-NK cells were used to treat tumors, and after they kill one specific type of tumor cells, other tumor cells may still proliferate, and probably leading to tumor recurrence. So to overcome those obstacles, dual-target CAR-NK cells and cytokine autocrine adapted cells have been engineered. However, these strategies will impose a burden on cells' protein synthesis, and thus new strategies are urgently needed. So to illustrate our design, consider this metaphor. If our NK cell is like a swordman, we need to not only make him stronger, but also give him multiple weapons to combat with different enemies. And such process in our experiment means the evolution on CAR-NK cells. And evolution is a long-term process where advantageous genes are preserved over generations, so nature can select the survival of the fittest. And in our experiment, diverse mutant clones are generated in laboratory and subjected to the artificial selection pressure, and just analogous to the environmental selection pressure in the natural evolution. So first, we want to solve the activation failure of CAR-NK. And here you can see the classic second-generation CAR structure. It can recognize the tumor ligands, then stimulating the CAR host cell. However, NK cells still lack the capabilities of proliferation and cytotoxicity due to the interleukin-2 deficit. And notably, the activation of the JAK-STAT downstream of the interleukin-2 receptor depends on the oligomerization of the beta and gamma chains. And experimentally, membrane proteins can oligomerize through the electrostatic interactions, such as NKp44, which facilitate activating signals by associating with negatively charged residues. So we hope to design a class of structure based on the charge-induced oligomerization. So here you can see we engineered classes of CARs configuration targeted CD19, and some is the tandem interleukin-2 receptor ICD CAR, incorporating the intracellular segments of the interleukin receptor beta and gamma and serial linked to CD3 and some are the charge-attracting interleukin-2 receptor ICD CAR, featuring a positively charged transmembrane region connecting 4-1BB or CD3 or a negatively charged transmembrane region linked to interleukin-2 receptor ICD. And each CAR is also fused with an EGFP for tracking. And then we do the selection and got 3 dominant sequences. Okay, and as for the second round evolution, considering the heterogeneity of the solid tumors, so here we replace the SCFV segments to the extracellular segments of the NKRs and facilitating the display of the NKR library. And such process is just equivalent to equipping the swordsman different swords to combat with different enemies. So overall, for the whole evolution process, we aim to solve the problems of cytokine deficiency and tumor heterogeneity, and enabling our NK cells to achieve the auto-expansion and the pan-cancer recognition. And now let's see our results. So first, we successfully established our library, And we found they can proliferate even without the external cytokines. And then we do the selection and got 3 dominant sequences. And we named them ECAR 1, 2, 3 respectively here. And we also designed ECARs with different target construction, including C1920, GPC3, and MSLN. And we found these ligands all can exhibit a great cell expansion and amazing activation of the signaling pathway of the JAK-STAT, and the great cytotoxic activities in vitro. Okay, and as for the in vivo experiments, so we successfully established NCG and NCG-15 mouse models. As for the in vivo image system, we found that in the cytokine sufficient condition, both FSC90 CAR and ECAR can restrain the tumor growth. However, in the cytokine deficient condition, only our library FSC90 ECAR can restrain the tumor growth and greatly extend the survival of tumor-bearing mice. And also we do the immunofluorescence, and we found our library can reduce the tumor burden and improve infiltration. Okay, and let's get to the second round evolution result. So here we utilized NKRs as sensors. And so since we have already converted the inhibitory signals into the activating signals, so utilizing KIRs as sensors also allows for the activation of NK cells. So in our experiment, it was found that different tumor stimuli could promote the proliferation of NK cells. And through the NGS, we found that the ACAR NKR can proliferate in response to different tumor antigen signals. And the NKG2D, such ligand, is a kind of all-for-one for all, which can recognize the pan-cancer. And let me briefly show you a result to how to understand such results. So we have already known that the corresponding ligand for the NKG2D are MICA, MICB, and ULBP1, 2, 3, 4, 5, 6. So we also know that these ligands are highly expressed on the Reggie cells. So we can use NKG2D to treat the Reggie cells. Okay, and we also co-cultured our library with the different tumor cell lines and also the PODX models to testify its anti-tumor efficiencies in vitro and in vivo, and they also showed higher anti-tumor efficiencies. Okay, and in conclusion, we got our ECAR library through 2 rounds of evolution. So we enable our NK cells to achieve the pan-cancer recognition and the ligand-dependent expansion through the charge attracting principles. Okay, and that's all of my presentation, and great thanks for everyone. Thank you very much.
Thank you for the interesting topic. Do you have any comments or any questions?
For your NK receptor library, so what is the criteria you use to select the NK receptor domain into your library?
NK receptor domains. Okay. Sorry. Let me turn my slides here. Maybe you mean ask for the NKRs we select here. So NKRs generally here we select these ligands here. So generally there are 2 types of NKRs. One type is the, so you can see here. Let me show it. Sorry. So here you can see that this is the non-HLA specific receptors, and one is the HLA1 specific receptors. So generally, in human bodies, many HLA1 receptors are highly expressed. So for the safety problems, we will generally use the non-HLA1 specific receptor here. That's for the safety. And these are the receptors already exist on the NK cells. So generally we divide them into 2 parts, and we select this one. And we incorporate these receptors into our sequences here. Thank you.
Is there a preference or specific to particular type of tumor, or this is a general marker?
Sorry, can I listen again?
I mean the NK receptors that you choose here. It is a general protein for all tumors, or it is specific or high expressed in particular type of tumor?
Oh, okay, so these are the receptors and these are the ligands expressed on the tumor cells. So different tumor cells will express different ligands. So this is NKR, so it's a whole library. So generally there are many kinds of receptors, and one NK cells will express one receptors. So, and as for the tumor cells, they will express different ligands. So when it comes to the clinical scenarios, we'll use these different NKRs to treat different tumor cells. So that's why we say it can recognize the pan cancer. Thank you.
Thank you for the interesting topic.